Hello again, everyone, and thank you for joining us as we discuss one more concept of physiology and anatomy. Today we will be talking about the immune system. We will be discussing the adaptive immune defensive. Just as a reminder of how the immune system works, we discussed the innate immunity, the first and the second line of defenses. We're going to go to the third line, which is the adaptive. And just as a quick reminder, the first and second line of defenses, the response is very immediate. The body is going to launch its defense very quickly. With the adaptive, it takes a little while because the immune system is being exposed, it's starting to recognize the antigens, and there's a little time for activation of the B and the T lymphocytes, as we will see. So when we talk about the adaptive immunity, there are two very important parts. Number one, they have the ability to recognize specific antigens. So while my innate immunity does not know what is invading my body, the adaptive or specific immunity has the ability of recognizing the specific antigen, and we will learn how this happens briefly. The second thing, it has the ability of keeping this invader in memory so that when there is a second attack by the same antigen, the adaptive immune response is going to be faster and more efficient. To understand this whole process, let's start talking about what's an antigen, what's an invader. So this is an antigen. It can be a whole cell, like a microbial cell, and most of the time it can be only a part of this cell. What is important about it is it will always have an area, and this area is called the epitope or the antigenic determinant, and this area is what is going to be recognized by our immune cells and what will activate those immune cells. And this is how the recognition occurs. So if this is the antigen, it has a specific epitope, and remember, epitopes take shapes, different shapes. This shape can only be recognized by an antigen receptor. So my immune cell will have an antigen receptor that can recognize the antigen by its specific shape. And then they can attach shape to shape because of this recognition. Where do the B cells and the T cells come from? So we were talking about the adaptive immune system. The cells that will react are the B cells and the T cells. Where do they come from? So the origin of both the B cells and the T cells is inside the bone marrow. They are immature cells. They will mature in different places. So the B cell will remain inside the bone marrow until it is completely mature. The T cell will leave the bone marrow and go to the thymus gland, where it will gain its maturity. Once the B cell is mature and the T cell is mature, they will go to the lymph nodes, they will become immunocompetent, and they will learn how to launch their attack. The B comes from bone. So the B cell is dependent on the bone marrow for its maturation. The T cell is dependent on the thymus gland for its maturation. We'll look at this process in a little bit more detail. So again, this is the bone marrow. This is the origin of both the B cells and the T cells. The T cell will go to the thymus for its maturation. The B cell will mature inside the bone marrow. They will go to the lymph nodes. They are going to be immunocompetent cells. They will learn how to attack antigens, and now they are going to circulate through every part of the body looking for those invaders to attack and destroy. So the two branches of the adaptive immunity. One branch is called the cell-mediated. The other arm is called the humoral immunity. We're going to look at differences, but we're going to remember that this will work together. The whole body works together. We're just learning the differences between them. Who is the main responder in my cell-mediated immunity? It's the T lymphocyte. And we have two types of T lymphocytes, the cytotoxic T and the helper T cell. Who is the responder in the humoral immunity? It is the B lymphocyte. How about the antigen? In the cell-mediated immunity, the T lymphocyte can only recognize antigens in the form of cells. It has to see a big cell, an infected cell, a cancer cell, or a transplanted cell, and it can attack it. The B lymphocyte cannot recognize cells. What it can recognize is small fragments of antigens outside the cell, mostly in body fluids. And this is where the word humoral comes from. Humor means fluid, so antigens in the body fluids are going to be recognized and attacked by the B lymphocyte. How about the process of antigen presentation? For the T cells to be effective, they need a cell to present the antigen for them. We call this the antigen presenting cell. And only if the antigen presenting cell presents the antigen, they will be activated. The B lymphocyte does not need this presentation. As soon as it finds the antigen, it will be activated. What's the final outcome? Once the T cell recognizes an antigen and becomes activated, the cytotoxic T will actually destroy this cell. We call this cytolysis of the infected or abnormal cell. How about the B lymphocyte? What is the effect of the B lymphocyte? The B lymphocyte will change into a plasma cell. The plasma cell secretes a huge number of antibodies against the detected antigen. And again, we're going to look at this concept and look at the details. So we're looking at the cell-mediated immunity versus the humoral immunity. T lymphocytes only recognize cells. The B lymphocytes recognize small antigens outside cells. The T lymphocytes are two types, the cytotoxic and the helper T cells. The B cell, we have only one type of B cell. The cytotoxic T cell, once activated, will destroy the abnormal cell. We call the cytolysis. The plasma cell is the product of a B lymphocyte, and it will secrete antibodies to destroy the antigen. We need to pay a lot of attention to the helper T cell here, because the helper T cell has a very important central role. We see the helper T cell secreting cytokines, which are some signaling activating proteins to activate the cytotoxic T. They do the same to activate the B lymphocyte. So the helper T cell has a central role in activating both sides or both arms of the immune system, the cell-mediated and the humoral immunity. Let's talk again about a little bit more detail of what occurs in both of those processes. We're going to look at the cytotoxic here. This is the T lymphocyte maturing in the thymus. As it comes out of the thymus, please remember, I have two different types of cells, T lymphocytes leaving the thymus, the cytotoxic T cell and the helper T cell. Coming out of the bone marrow, the mature B lymphocyte, I have only one type of B cell. Let's look at the surface of those different lymphocytes. Each one of them has an antigen receptor on its surface. So the cytotoxic T has an antigen receptor, the helper has an antigen receptor, the B cell has an antigen receptor. What is the function of the antigen receptor? It will identify and recognize one specific antigen. Something else, when we look at the different T cells, each cell has a certain protein on its surface. The cytotoxic T has a protein called the CD8. The helper has a different protein called the CD4. We will look at these and see that they identify the cell, but they also have a function to do. And again, the process, the activation with the help of the helper T will result in cytolysis, in cell-mediated immunity, production of antibodies in humoral immunity. We will look at the antigen receptors and the CD4 and CD8 proteins to understand these. These are antigen receptors. So this is a B cell with antigen receptors on its surface. This is a T cell with antigen receptors on its surface. What is the function of the antigen receptor? To identify one specific antigen. How do they do that? The antigen receptor has a shape and it will only recognize an antigen that is complementary to this shape. Again, there's going to be one shape here. This is where the tip of the receptor is, and this B cell can only recognize one antigen which has the shape over here. So an important thing to remember, each B and T cell has one unique set of antigen receptors, and accordingly they can only recognize one unique antigen which has the complementary shape. How about the CD4 and the CD8 cells? This is the helper T cell. When we look on its surface, it has a very specific protein. This protein is called CD4 protein, and accordingly, helper T cell is called the CD4 cell. When we look at the CD8, it has another protein on its surface. This is called CD8 protein, so the cytotoxic T cell is called CD8 positive protein cell. What is the function? They identify the cell, but they will also have a very important function in recognizing specific antigens, as we will see in a minute. All right. To understand the process of activation, we have to look at a few more things. This is a little summary, so we're not confused. Cytotoxic T cell is called the CD8 positive cell. It will kill infected cells by cytolysis. CD4 is a helper that will help both the CD8 and the B lymphocyte. The B cell is one that will produce the antibodies. Another concept to help us get all of this story together is the MHC proteins. What are MHC proteins? They are self-marker proteins. Where are they located? On the surface of every cell in our body. Let's look at them. So, if this is an epithelial cell, a muscle cell, a white blood cell, a leukocyte, or a nerve cell, every nucleated cell in our body will have an MHC protein. What does it do? It is a self-marker protein. What do I mean by a self-marker? It has to be present on the surface of my body cells so that when the immune cells come and they are checking the cell, they can see the self marker and they will not attack my own body cells. So their function is for self-recognition, to help the B cells and the T cells know this belongs to my body. We will not attack it. So the function of MHC protein is, number one, self-recognition, to prevent my immune cells from attacking my own body cells. Now remember, every person has their own unique MHC proteins, which are very different from any other person, except, of course, in identical twins. What else is the function of MHC proteins? They will take a role in presenting the antigens, which we will look at in a minute. So they can take an antigen, attach it, and present it on the surface for immune cells to come and know, now we need to come here and help. There is an attack. Now, are there more than one MHC proteins? Yes, there are two. We call them class 1 and class 2. This is the story of antigen presentation. So if I have an antigen that has invaded my body, it has broken the first line of defense, the second line of defense, and now it is inside the body. A phagocytic cell is going to take this antigen, is going to break it down. It will process some of the parts, get rid of some, and it's going to take a very specific part of the antigen and present it on the surface. Guess what? It has the epitope. So the MHC protein belonging to this antigen presenting cell will present the epitope on the surface. It processed the antigen, it took a certain part and presented it. And now when the immune cells, B cells and T cells see this presentation, they will be activated, they will be attracted, they know they have a role to come and attack this antigen. So we call this the antigen presentation. Let's look at the cytotoxic T and let's look at the helper T, what is presented to them. So when we look here, if this is an infected cell, the infected cell is going to take an antigen, break it down, fragment it, process it, and then this is the MHC. It's going to take part, a very specific part, the epitope of the antigen, and present it on the surface. Cytotoxic T that is circulating is going to be attracted and recognize this is an antigen. And now cytotoxic T is going to be activated. We will learn the process later on. So MHC proteins are presenting the antigen so that the cytotoxic T will recognize it needs to play a role over here. The same for helper T. This is again another cell. This is an antigen presenting cell. It's going to take in a microbe, fragment it, process it, and this is the MHC. It will pick up a part, which is the epitope, and present it on the surface. And now the helper T cell is going to be attracted to this area because it recognized there's an antigen and it has a role to play. There are two classes of MHC protein, MHC1 and MHC2. If the antigen is presented on MHC1, cytotoxic T is going to be attracted and will recognize it has a role over here. If the antigen is presented on MHC class 2, the helper T CG4 cell is the one that's going to be attracted and launch the attack. So there's a little different role for the CG4 and the CG8 cells. And this is a little summary on the MHC protein. So MHC class 1 is present on all the nucleated body cells. So the only cell in our body that does not have MHC1 protein is the red blood cell. This is why the red blood cell can be destroyed by phagocytic after a lifespan in the blood. MHC class 2 is on the surface of all antigen-presenting cells. Macrophages and dendritic cells are examples of those cells. The presentation on MHC class 1 is going to be recognized by the CD8 or the cytotoxic T cell. It's going to be attracted, launch its attack. This on MHC2, the helper T cell is who recognizes and will launch the attack. Let's put all of this story together. We're going to look at the humoral immunity, look at the steps and how antibodies are formed, and then we're going to look at the cytotoxic on its own and see how cytolysis will occur. So with the humoral immune response, these are the steps. This is the B cell. The B cell meets small particles of an antigen that are outside cells. The specific B cell that can attach to this antigen, remember, has a very specific antigen receptor. The complementary shape helps it recognize the receptor. So this B cell cannot recognize, this B cell will not recognize, only the one with the complementary shape will recognize the antigen and attach to it. Once it attaches to it, the helper T cell will come along. Why? Because now the B cell has processed part of the antigen, is going to present it on MHC class 2. There's the antigen, and now the helper T cell is attracted. It knows it needs to come and stimulate the B cell. What is the effect of helper T cell? Helper T cells produce certain signaling or activating proteins. We will collectively call these cytokines, and the cytokines will signal to the B cells or later the cytotoxic T and stimulate them to start being activated. So what we see here is helper T cell arrives, it attaches, and it starts producing those interleukins or cytokines to activate the B cell. What happens to the B cell once it's activated? It's going to divide, make many copies of itself. We call these clones. So the B cell will divide and make clones. Most of these clones are going to differentiate into the plasma cell, and this plasma cell will produce the antibodies. A few of them will not change into plasma cells, but they will become memory B cells. What is the function of the plasma cells? Plasma cells produce, they will secrete a huge number of antibodies. All of those antibodies have that specific shape. They are going to attach to this initial antigen. They are going to inactivate it. We're going to see how. And they are going to finish it up. They are going to overcome the infection. The memory B cells will not attack this antigen. They will remain in our system for a very long time. After all of the antibodies have gone away, they have done their work, this memory B cell will remain in our body for many years, so that if there is another infection with the same antigen, these memory B cells will act very quickly. We don't have to go through all of those stages. They will act very quickly, recognize the antigen, produce antibodies, and destroy the antigen much faster and more efficient. So accordingly, the first time the body is invaded by an antigen, we call this the primary immune response. So this is the primary response. This is the first time. It's going to take a longer process to do all of this. So the primary response, we have certain antibodies that are being produced. The IgM antibodies are the ones that are mainly produced first, and then the IgG. My main responder here is the plasma cell, as it makes the antibodies. In the secondary response, can you see in a very short time we have a huge number of antibodies that will totally overcome the antigen and destroy it, and the effector cell here is my memory B cell. It produces antibodies, and in this case it's a different type of antibody. IgG antibodies are produced a lot more and faster than IgM. What do antibodies actually do? How can they overcome the antigen? So we're going to briefly look at the shape and the different classes of antibodies. This is the shape of antibodies. They are made of long or short polypeptide chains. We call these the heavy chains. The longer ones, we call these the light chains. So 4 polypeptide chains. Every 1 unit of antibody has 2 antigen binding sites, and you can see the shape of the site as it is going to recognize one specific antigen. We will also remember that there is a complement binding site here where complement can attach to the antibody. What is the function? What are antibodies actually going to do to this little antigen? They can do a variety of things. We're going to talk about these. They can just cover the area, one specific area that is the attacking part of the antigen. If they do that, they will mask this specific area, and we call this neutralization. They're going to come and cover up and mask this specifically dangerous area of the antigen, so it will not attack the body. We call this neutralization. If they are able to attach several of the antigens at one time, so all of these are antibodies, that's a big one, they will form one big clump, and we call this agglutination. If they're attaching to a smaller particle, we call this precipitation. All of this will lead to more phagocytosis. So all of these are going to be picked up by the phagocytic cells and destroyed. They can also do what we call complement fixation. So if you remember when we spoke about complement, we said that complement can be activated and form the big MAC, and MAC will poke holes into a cell and cause cell lysis. So this is the area where complement can attach to the antibodies and be activated and cause cell lysis. All right. Is there only one class of antibodies, or are there more than one? There are more. There are 5 classes of antibodies. Immune globulin is the same for an antibody, and they take the letters G, E, D, M, and A. They can be either a monomer, a dimer, or a pentamer. They can be 5 molecules attached together. Very briefly, we're going to look at the specific functions. There are many more functions. We're going to look at 1 or 2 specific functions of each of those classes. So IgA is present mainly in the mucous membranes and in the secretions of the mucous membranes. So we find it in tears, in saliva, in the mucous membranes of the stomach, and so forth. What do? They adhere to the antigens and prevent them from penetrating and going into the body tissues. IgD is the antigen receptor on the surface of the B cell. it always attaches to the surface of the B cell and it helps the B cell to be activated. So if I were to go back here and look for a B cell, this is the IgG. It is the antigen receptor that sits on the surface of the B cell. So it takes part in the process of the B cell activation. IgE is mostly on the basophils or the mast cells. It activates them to break up and release their histamine, so they play an important role in inflammation, and they also help attract eosinophil towards parasites to destroy them. IgG is by far the most common. It's about 80% of all our circulating antibodies, and one of its very vital functions is it is so small and it can pass through the umbilical cord from the mother to the fetus to give the fetus passive immunity until it is born and for a few months later. IgM is the largest. It's usually in a pentamer. So if I have 5 molecules, this has 10 binding sites for an antigen. So it really can bind lots of antigen and is effective in agglutination and complement fixation. How do we become immune? How do we acquire immunity? We can do this by 2 ways, either actively or passively. We acquire immunity actively if my body is invaded with an antigen and my own B cells are being activated. My own plasma cells will produce my own antibodies. So my body is actively involved in producing the antibodies. Passively, I am not producing the antibodies, but I am receiving them from another source passively. I don't take part. Let's see examples of active immunity. This can occur naturally or artificially. Naturally, if we are infected, if we get sick. So if I am infected with the measles, my body is going to be activated. I'm going to make antibodies against the measles and I am going to be immune against the measles. So this is natural. Artificial is by means of vaccination. So when we want people to get immunity against certain diseases, we will inject a vaccine and activate their system. Passively, again, this can happen naturally when antibodies are transferred from a mother to her fetus. The baby will passively receive the antibodies through the umbilical cord or through the breast milk. Artificially, when we inject people with antibody serum to get them immunity very fast if they are exposed to an infecting agent. So this is about humoral immunity. We are going to talk about the cell-mediated immune response. So who is responsible? The T lymphocyte. How many types of T lymphocytes do we have? We spoke about the helper T or the CD4 and how it helps both the cell-mediated and the immunity. We spoke about the cytotoxic. We're going to describe its role in a minute. There's going to be a memory T cell and a regulatory T cell. So let's look at the process here. We're talking about the helper T cell. How does the helper T cell get to be activated? Well, there's going to be an antigen presenting cell. It first takes the antigen, it will process it, it will present it on MHC class 2 protein. Once the antigen is presented, CD4 is going to be attracted to the area, it's going to attach to the antigen, it's going to make many copies of itself, make clones of itself, And most of those clones are now activated, and they are going to go and help the B lymphocytes or help the cytotoxic to launch an attack on this antigen. Some of them will not be activated helper T cells. They will become memory B cells, the same idea. So if there is another attack with the same antigen, the memory T cells are going to be activated a lot faster and launch the immune attack. This is what the helper T cell does. So when the helper T cell recognizes the antigen, becomes activated, it will secrete cytokines. They are signaling proteins. They will signal to the cytotoxic T cell activated. They will signal to the B cell and activated. What is the cytotoxic T cell going to do? So again, this is the cytotoxic T cell. When it is presented with an antigen on MHC1, it will be attracted. It will come to this area. And remember, it's not on this diagram, but it will receive signaling from helper T cell. Once it receives this signaling or co-stimulation, the cytotoxic T cell is going to multiply, is going to divide, and is going to be activated, and now it has the ability of actually destroying cells. We're going to look at this in a minute. Some of those cells are going to become memory T cells for the incoming attack. How do these activated cytotoxic T cells actually destroy an invading cell? This is what they do. They will come and they will secrete two very specific chemicals over here. One is called perforin, and perforin comes from perforations. So they will attach to the abnormal cell and they will poke holes in it by the perforin, and then they will secrete granzymes, and granzymes will enter through the pores and they will digest this abnormal cell and destroy it completely. So this is the effect of the cytotoxic T. So we spoke about the helper T, the cytotoxic T, the memory T. The last T cell we're going to talk about is the regulatory T cell. What does the regulatory T cell do? It regulates the function of the immune system. So once the attack is complete, once we destroy the antigens, the regulatory T cell tells the immune system, the war is over, there's no more activity, we don't have to keep launching the effect. So it prevents the immune system from overreacting. This is the function of the regulatory T. This concludes our lecture for today, and thank you for being with us.